Hey guys, welcome back. This is just wanted to kind of go through an applied proteome analysis here. This is liver cancer, and it's just a paper that was published 2014, and it's their discussion about hepatocellular carcinoma, and it's a review paper, and it talks about proteomic techniques to answer clinical questions. And this is a pretty good paper. It looks at studying cell lines, animal models, human tissue, bodily fluids from patients. So it's a very broad approach using proteomics to look at all these sources of information here. So you should know by now that, starting out, proteins have to be isolated from some sample. That could be from the patients, from the model systems, from the cell lines, from the animal models. That means those are mice or whatnot that actually have the disease. And then that will potentially fractionate the proteins further, and that can actually enrich certain proteins, such as post-translationally modified proteins. And then the paper discusses some strategies going from top down or bottom up approaches. And this is showing in this figure, this is patient samples. Here's the tissue, bodily fluids, and we're isolating the proteins there. And we get some type of fractionation, looking for different proteins. Proteins that are in abundance, you might want to get rid of. This may be looking at proteins that are rare here. Here's model systems, animals, cell lines, and again, protein isolation, and then focus fractionation. And so the bottom-up experiments, the protein samples are digested and separated using the maltitol for the electron spray or something called tandem mass spec, and proteins are identified. So this is kind of a bottom-up approach. And then a top-down strategy is where one uses two-dimensional gel differential electrophoresis to separate proteins and digest it here. So this is top-down. You have the protein mixture. You do your 2D gel. You make your peptides, and you got the mass spec. And you can also go get from a protein mixture and peptides of all proteins. Okay, so all proteins available. This is focused proteins right here on a 2D gel. Okay, so there's a focus here. This is more, we're going down and we're looking at proteins we suspect are an issue. Here it looks like you're looking at all proteins, and then you might do chromatography and mass spec. So these would, the top down would find proteins that are predicted or there's evidence for an effect. The bottom up is you assuming you don't know, and you don't want to miss anything that nobody's predicted before. So kind of that's a subtle difference, but it's a big difference here. Again, you need what people think is there, and then you need to find the unknown unknowns here. What people don't know is there, right? It's you don't know what you don't know, and that would be in this area. And so we're looking at all proteins. Either way, you're doing the protein identification by the sequencing that we've talked about, Edmonds or the maltitol for the other mass spec here. And so here is just showing some examples of cellular models, animal models, body fluid, and tissues, showing the procedures that were used. Some of these we've talked about. the type of mass spec that was used, and the clinical questions here. Here's looking for biomarkers, changes in, I don't know what AFP expression is, but that must be something related to cancer. And so this is, certainly beyond the scope of this course, but all the references in your that paper, you could actually follow up on that. So you see right away that we're getting lots of different information and putting it together here. So quantification strategies, we identify as many proteins as possible. The accurate quantification is a major goal. So they're looking for key biomolecules and the methodologies here to monitor relevant or absolute changes. So we're looking at what's going up, what's going down, and the different depends on the amount of samples. So we didn't really talk about that, but certain techniques require a certain amount of like biopsy tissue or fluids, and others like cell lines, you could probably grow vats and vats of it. And each different approach will have positives and negatives here. And in these kind of experiments, you're going to have to do multiple times. In other words, you don't want to just make conclusions based on one roll through it. You're trying to figure out what's going on. You're quantifying the advantages. How accurate are these experiments, right? How much of the proteome is covered? In other words, are you covering everything? And of course, efforts and costs. So this is kind of a broad brush approach in this paper's review, looking at all these different experiments from different groups that are coming together. And this review paper actually puts it into place. And this is just showing applications of proteomics here. So the Seldi chip is what we're going to go next. It's a big technique that allows high throughput data very quickly, you'll see. And the nature of the experiments with Seldi chips rely on things like affinity chromatography. And so we'll see how that chip has scaled down that concept to grab certain proteins for further. A company named Cyphergen developed this probably about 5 years ago. And what they were able to do would be to purify and actually enrich certain proteins on the surface of their chip. And so the chip would preferentially bind. So think about the affinity, things like charge, hydrophobic, hydrophilic, other kinds of maybe some lectins or metal binding, those kind of things. So think of all the different characteristics that we use for affinity chromatography of proteins. They've scaled it down onto a small postage size stamp, maybe bigger than that, but it's not too much bigger. And so what they've added to the chip was UV absorbing matrix, and then it's placed into a mass spec. So they're grabbing the proteins, having a matrix, exciting it, and then sending it to the mass spec through the laser. It's not just a light, it's a laser, firing of a laser at the sample matrix mixture. And then the mass charge of the proteins in that sample. And then you use computer analysis to look for differences, right? So like unaffected patients and affected patients, cancer, non-cancer, that kind of thing. And so what the Seldi chip does is they've set up grids on the chip that preferentially bind to certain proteins types. and that's the key thing. It's called surface enhanced laser deabsorption ionization, Seldi. Okay, so these are the Seldi protein chips. Okay, each chip binds a different class of proteins, and the matrix is here, and you hit this spot with a wavelength of light, and here it is here. So your sample goes directly into the array, and they called it array, like a bingo card. Proteins are captured. The ones that are not captured are washed off. So it's an affinity capture. And the matrix is added to the chip, and then you hit the laser that excites the spot, and you measure the mass charge of what protein stuck. Okay, so looks like the red protein stuck. I don't know. They did. So here's a spot that does reverse phase. This is hydrophobic proteins, and membrane bound proteins will be snagged there. Okay, think of these as baits, right? So the proteins will bind the reverse phase. Metal ions and phosphoproteins will bind at this spot, ion exchange and cation exchange. So now we have specifically divided multiple baits in the protein mix, and we grab them. Okay, and so this is about the size of it. Okay, and Biorad is the company that owns Cyphergen, and here's an 8-spot array. You can imagine you could do multiple spots, even more than 8. And each spot is preferential to the type of proteins that might be in the sample. So you can see right away we've already divided out from a mixture proteins. So we've affinity bound certain proteins with properties, and then we can study those further and figure them out. So selective proteins of interest. And so the spots have different patented chemistries and are activated by the laser. And so that's the patent side of it. So you can mini fractionate on that one chip proteins in a mixture, types of proteins on the chip. And then you can figure out what the proteins are. So here's another cartoon showing different spots here. So any surface chemistry that you can think of and put onto a spot, you could add to a Seldi protein chip. And each chip is analyzed by itself, so we don't get any cross reaction here. So you're saying, well, what of a hydrophobic positive charge? Probably and again, they would know this, but one would probably trump the other. So all hydrophobic may bind, and so hydrophilic positive would bind and hydrophilic negative might bind there, that kind of thing. So the UV absorbing matrix is also on the chip, and is you hit those, make the charged ions, right? You got a charge mass ratio. And here's the chemical, not that you have to memorize that. And you hit with a UV laser pulse 4 nanoseconds. And that matrix allows a proton to be added from the peptide or protein, and it causes it to be charged and ionized. And the charged proteins are accelerated through a series of different plates and travel through the mass spec in the machine. Okay, so the firing hits the sample, the little spot, and then you're doing this MALDI, TOF, or other kinds of mass spec analysis here. So you have your samples, you wash, you add the absorption matrix that is sensitive to the laser. You then turn it over and you put it in the machine. Here's the laser, and here's your time of flight tube here. And here are the ions from spot 4 coming down and they're being detected here. And so you're getting in this cartoon 3 proteins here of different, this is the raw spectra, and then they can actually do a grayscale view of it and figure out the amount. So you got the amount, right, the intensity, and the charge here, and the mass. So in cancer, what does the profile look like? Non-cancer, what does it look like? So differences will be detected right away. Of course, for informatics, all this information is stored and further patient sample can be compared. So here it is, the reader, and the reason they multi-spotted it was it just makes it simpler. Okay, so if you put too many proteins on a spot, right, you might suppress the ionization. So this separates proteins out and makes it more accurate as to what is in the spot here. And so it's talking about low abundance proteins. Modified proteins might be hidden or underestimated. So, we're talking a biomarker, but you're missing how abundant the biomarker is, or it's hidden and you missed it. So that's part of the multi-spot purpose here. And of course, there's the ionization there. And again, we use the multi-TOF as one to figure out the masses and figuring out the time it takes to travel down the flight tube. And that's proportional to the square root of its mass. And so now they know the mass of the fragment or the protein here. And that's the flight tube coming down there, time flight. And so analysis of groups of samples can be looked at over time, condition, diseases. So this is a biomarker software. They keep track of all these profiles, and they can be then accessed and compared in a disease versus a control, or maybe disease versus a treated versus an untreated controls. And they cluster, they actually find the different proteins and see the different distributions of, or it's called differential protein expression here. So you can ask the question, what's different about multiple samples here? It's really kind of neat. So this is a real high throughput approach. So each little disk can be a patient samples there. And then of course they've been software that's developed, and so this is proteomics here, and we get a spectrum for each spot. You get the marker to find things that are different or in one type of situation versus the other. And you get the mass charge and the statistical value. In other words, how many times do you repeat this and still find that spot? Okay, so is this real? Can we trust this data? Okay, so you have to repeat it and probably maybe do even multi uh, uh, samples from patient. And look, they divided the chip controls versus treatment, probably different spots, and compare the controls versus the treatments here. So we can look at proteins interacting with other proteins or DNA metabolites. In other words, you can put DNA on those spots, you could put RNA, you could load it with certain proteins to find protein-protein interaction. And here's an example. I want to show a clinical, and it shows the purification of a compound known as transferrin. Don't know if you know ferrin. Ferric is iron, and so our bodies actually will take in iron, and they take it in by a receptor called the transferrin receptor that will grab it from outside and release it inside the cell and go back and grab some more. And it looks like certain conditions, certain diseases, that the transferrin receptor might be different. And so an example of one of these diseases is called the CGD, or chronic granulomatous disease, Bridges-Good syndrome, Quay syndrome, chronic granulomatous disorder. These are genetic diseases of the immune system, and the immune system cannot make reactive oxygen compounds such as superoxide. Superoxide, if you're from microbiology, kills bacteria. And so if you don't have superoxide, that's going to build up these bacteria inside, and that becomes a granuloma in the organs. So you can imagine big hunks of bacteria in your organs are not going to be good for the organ function. And it was found in 4 boys in 1950. And it's a childhood disease. And they found some, and it's X-linked. So it's an X-linked form is one. So they've been working on disease for quite some time. And so here's the normal transferrin molecule, and there are our asparaginins. And there's the sugar trees. These are the high complex sugar trees. So it's gone through the trans-Golgi here. And you can have a spot on one of these chips that has an anti-transferrin antibody. So we'll pick these up. So you can take 1 microliter of patient's plasma, and of course there's other proteins, but there's a transferrin as well. And you incubate for an hour, then you wash off. And there is the transferrin you captured, and you wash off the serum albumin and other kinds of proteins. You add a matrix, and you hit it through the old time of flight here, that spot that you just grabbed the transferrin, and you get a reading, charge mass ratio. Okay. Flight of the ion there it is in green. So it turns out that in normal patients, which we are, we have control plasma. When they do this and look at transferrin, all our transferrin molecules have 2 carbohydrates attached to it. However, and that's called diglycosylated transferrin, they have found in this disease mono and unglycosylated. So when they add a patient sample from the CDG patients and do this spot and do the analysis, look what they find. They find a kind of a shoulder peak there, a peak here, and a peak there. This is unglycosylated here. This is monoglycosylated there, and this is the normal diglycosylated, which right there. Okay, so now we have a way, if an infant, if there's something they suspect, they can run their plasma and they would find unglycosylated or monoglycosylated, and that would be a biomarker for this disease. And they can then, scan with one of these chips as many spots as they can put together there. I mean, if it's 8 spots, you could do 8 plasma samples. You might do 4 patients twice to, confirm what you're finding here on your time of flight, having these informations here. Okay, so again, the reason the unglycosylated's first is because it's lighter than having 1 sugar tree, and the heaviest would have 2 sugar trees. Yep. And so that's my 10-cent tour of the Seldi chip and some of the mass spec here. So we'll end this lecture here. So come back, we will talk about protein interaction technologies, and we'll have lots of fun there. Okay.